Hello. So good morning, everyone, or good afternoon, or good evening for Linda. So we are pleased today to have on the Worldwide Neuro today Linda Richards from Brisbane. So just for those of you who don't know Linda, Linda has been professor at the Queensland Brain Institute in Australia for about 15 years. Before that, she studied in Australia, but also did a postdoc in the US and had the first lab in Baltimore before moving to Australia. So what also some of you may know is that she was appointed recently to St. Louis in Missouri, so where she will be director of the McDonald Center for Cellular and Molecular Neurobiology at Washington University School of Medicine. So maybe she will say something about this new appointment. that looks exciting. So Linda worked for many years now on the development of the corpus callosum and axon guidance, and she will tell you more today about this. She was also, she's a past president of the Australasian Neuroscience Society. She was also much involved in the BrainBee Challenge contest. That is a worldwide event now. There will be one in France in 2022 for the fans, so get ready for it. She was also one of the founder and someone who pushed a lot to have this IRC5 consortium, which is the International Research Consortium for the Corpus Callosum and Cellular Connectivity, IRC5. You can look at it on the web. You can also try to become a member. So they have also regular seminars. It's also something that is very interesting. So I don't want to lose too much time. So thanks a lot, Linda, for attending. It's 8 p.m. for you, I think. So you had a long day. And thanks to all of you for being present. And as I remind you, you can ask questions. So in the bottom panel of your screen, you have the Ask a Question part. You can ask questions. You can also vote for questions. If there's one you really like, you can vote. It will be on the top. And then at the end of the talk, I will read the questions to Linda, and then she will answer them. So now, Linda, you can share your screen, and you're on.
Okay. Thank you very much, Alain. Share my screen and the presentation. You can see that, Alain?
Perfect. No problem.
All right. Well, thank you, everybody. It's really a pleasure to be with you wherever you are around the world. What an exciting seminar series. And thank you so much for Denis Jabadon and Alain Chaudetel for inviting me to be part of this seminar series. I'm going to talk today about my favorite topic, which is the corpus callosum and cortical development. And our lab is very interested in how the brain is formed during development to wire up correctly so that it can function. And what you're seeing here on the left is a looping video of a human brain, an adult human brain that we've scanned with our 7 Tesla MRI scanner at UQ. And you can see we have performed tractography on this diffusion imaging data, and you can see the exquisite connectivity throughout the brain. And our lab is very interested in how these long-range connections are formed during development and then how they underpin function. And our research is funded by the Australian government as well as the NIH. So as I said, we're very interested in these very early events in brain development that help to establish the circuitry of the brain, but then also how that circuitry underlies cognitive function. And the model system that we study is the corpus callosum, which is the largest fiber tract in the brain and connects neurons in the left and right cerebral hemispheres. So you can see it here. It's a huge fiber tract, very visible by MRI. And this is an adult human brain with the dorsal region dissected off, so you can see this massive fiber tract. Now, an interesting fact about the corpus callosum is that it evolved only in placental mammals, so such as humans, mice, etc., but did not evolve in other mammalian species, such as the marsupials and monotremes, even though these species also have a 6-layered cerebral cortex. And members of my group are very interested in how the corpus callosum evolved and the mechanisms involved in that evolution. And later in the talk, I'll be mentioning some of our work on this little guy, the Australian fat-tailed dunnart, and some interesting work that we're doing in this species. So just as by way of background, there are a number of different events that go into forming a corpus callosum, and these can be broadly defined in these 5 sort of stages. Firstly, the generation of different neuronal types, which so many people in the field have contributed to understanding both the layering and the transcriptional program of different cortical neuron types. And these projection neurons, of course, can be ipsilaterally projecting, contralaterally projecting across the corpus callosum, or various descending projections to the thalamus and also down the spinal cord and to other areas. There's also, of course, this specification of this projection. Midline crossing is an important step. I'll talk quite a bit about that today. But also target location in the contralateral hemisphere, and then innovation of the primary target and refinement of those projections as the circuit really wires up. So there's 3 main aspects of the work in our group. We work with human research participants, both adults and children. We also work on human fetal brain, fixed human fetal brain, to look at the mechanisms involved in formation of the corpus callosum. And then we try to understand the genes involved in callosal formation using mouse as a model system to look at the cellular molecular mechanisms of brain wiring. And more recently, we've been using the fat-tailed dunnart to look at activity-dependent mechanisms of brain wiring. So when you think about corpus callosum, you probably first think of the split brain patients that were studied by Roger Sperry, and for which he won the Nobel Prize. And these patients that he studied had their corpus callosum cut because they had very severe epilepsy. And he discovered that one of the major functions of the corpus callosum is to integrate information, particularly information that's lateralized within the 2 cerebral hemispheres. My group studies congenital malformations of the corpus callosum, and these people have very, very different phenotypes to people that have had their corpus callosum cut, such as split brain. So there are various disorders of the corpus callosum. You're seeing one here. This is complete agenesis of the corpus callosum. And in this person, the corpus callosum has completely failed. The callosal axons have completely failed to cross the midline. And so this is the neurotypical control, both in sagittal and in coronal view. And you can see that the axons have reached the midline, but fail to cross here. And so this occurs in around 1 in 4,000 live births, but it's also associated with around 300 different congenital syndromes. So that's where the person has a corpus callosum malformation, but also another malformation in perhaps a different organ system or another area of the brain. But there are some individuals that have corpus callosum malformation in isolation. And these people can be fairly normal functioning. Many people with corpus callosum dysgenesis have sensory or motor function difficulties. They may have delays in language development, developmental delay or intellectual disability, or problems with social interactions. And these symptoms have been beautifully outlined in a paper by our colleagues Lynn Paul and Warren Brown at Caltech and the Fuller in LA, where they have described this syndrome the neuropsychological syndrome of agenesis of the corpus callosum. And what they describe are three main core symptoms: slower cognitive processing, this limited interhemispheric transfer of complex sensory motor information, and also this restricted processing of complex and novel tasks. And these three core symptoms really lead to these more evident deficits in perhaps learning, memory retrieval, and this one, which I mentioned before, social interaction problems. So as Elaine mentioned, in 2015, together with other colleagues all around the world, we established an international research consortium called the IRC5. The C is Consortium for Corpus Callosum and Cerebral Connectivity. And here, our goal is to be able to pull data across these different sites from participants involved in our research. And this has really been fantastic for helping to power our genetic studies, but also imaging and neuropsychological assessments. So in our cohort within Australia, we collect blood and other biologicals for genetic analysis. And this work, I have been collaborating with Professor Elliot Scher from UCSF and also Bill Dobbins from University of Washington in Seattle for a number of years. And we recently sequenced a very large cohort of participants' families, trios, through the Broad Institute, over 700 families. So we're getting ready to publish that data, which is extremely exciting and really helping us to understand more about the mechanisms involved. In my group, we conduct multimodal MRI on the participants, a variety of different MRI sequences. And I also collaborate with Gail Robinson at UQ in the neuropsychological assessments of our participants. And from here, my lab takes this information and we use animal models to really try and understand the mechanisms involved. So just a little bit about the incredible plasticity that we see in these individuals and how exciting it is to work with these research participants. So here what you can see is a number of different participants. So here's a neurotypical control, and we're looking in sagittal view here. So somebody with complete agenesis. So dysgenesis is of the corpus callosum. Dysgenesis is a sort of umbrella term. Agenesis means no fibers crossing, and partial agenesis means some fibers crossing. But we're moving more towards the term corpus callosum dysgenesis because there's also people with thin corpus callosum and thick corpus callosum. So there's a lot of different varieties here. So in terms of these individuals, what you're seeing looking down on top of the brain in a horizontal view, we've color-coded the callosal axons in red here because they're running from left to right. And the axon tracks, these streamlines that represent axon tracks that are running from front to back are color-coded in green. So what you can see in the person with complete agenesis of the corpus callosum is that the fibers are not crossing, and instead they are forming these ectopic longitudinal bundles, which are known as Probst bundles because they were discovered by Probst in 1901. And here you can see a person with a very small remnant of the corpus callosum still. And we see across the different participants that you can have all sorts of varieties, every one that you can think of, in fact, we've observed, from very, very tiny remnant to almost a full corpus callosum. So this person who has a tiny remnant, you can see the fibers crossing here. So they also have Probst bundles for the axons that are not crossing. Now, more recently, work by Fernanda Tovar-Moll and Roberto Lent in Brazil, and also Elliot Scher's group, found that there are additional ectopic projections. This one, which they called the sigmoid bundle, and you can see why, is an ectopic projection that occurs in people with partial agenesis of the corpus callosum and is a connection from the frontal areas in one hemisphere to the occipital areas in the contralateral hemisphere. So normally, most of the callosal connections are homotopic. This one is really an ectopic heterotopic connection. Fernanda's group, interestingly, has also conducted tractography on her research participants in Rio de Janeiro, and has identified using tractography that perhaps some of the fibers actually cross via the anterior commissure. So when they're unable to cross at the midline, and people with perhaps complete or partial agenesis, they find other routes, either the anterior commissure or even indeed the posterior commissure she identified. This is an area of active research, indeed within the IRC5, an area of active debate, and you know, we are very keen to validate this kind of tractography in animal models. So hopefully from that, I've convinced you that the brain is capable of long-range axonal plasticity during development. So we're very familiar with synaptic plasticity. This is a different kind of plasticity which occurs during development. And let me just show you some further evidence of that. So this is our research cohort from Brisbane. And here, this is our cohort of people, some of our people with partial agenesis of the corpus callosum. These are sagittal views here and the coronal view here. And this work was conducted by Tim Edwards, who was an MD PhD student in my group and has been doing a postdoc at University College London, and also Dr. Ryan Dean, who's also a postdoc in my group. So what you can see here is a little schematic of the callosal remnant across 7 different research participants. And this is the tractography analysis, 3 panels for each participant, just different views. And although this is a very busy slide, what you can see from it is that even participants, for example, participant 1, 5, and 7, have callosal remnants which are fairly similar, of fairly similar size, yet they really do have quite vastly different connectivity between the hemispheres. So this is participant 1 and participant 5 is here. So this looks quite different to that. And this is participant 7, who looks really quite different to these two as well. So through this remnant, both our group and other groups within the consortium have found that even though the remnant might look very similar by mid-sagittal MRI in anatomical imaging, using tractography, it appears that there's a huge amount of variability of which areas are connected in each hemisphere. So, during his MD PhD in my group, Tim was also co-supervised by Elliot Scher, and he used a mouse model that Elliot's group had been using to try and understand more about this plasticity and connectivity. And so Elliot had previously observed that if you take a BTBR mouse, so this is a mouse that has a naturally occurring complete agenesis of the corpus callosum. It's a mouse that is commonly used in autism research because Jackie Crawley had identified actually that they do have a social interaction deficit. And if you take these mice and cross them with a black 6 mouse, and then you take the progeny from that and back cross it to the BTBR, you get an N2 generation where all the pups in the litter have different callosal phenotypes. So you can have pups in the same litter that have a full corpus callosum, a partial agenesis, or complete agenesis. And this provides a really nice mix of different phenotypes to study plasticity. And so here is Tim's cohort of mice that have, this is a measure of the amount of corpus callosum that the different mice in his group have. And this is just a color map of MRI imaging using our 16.4 Tesla small bore rodent magnet at UQ. And so here's an example of the full corpus callosum, partial ACC, and complete agenesis. And you'll notice there's some other red tracks here. These are the other major forebrain commissures, the hippocampal commissure and the anterior commissure. And so using this model, what Tim was able to do was to investigate whether mice also have a sigmoid bundle. That is that ectopic bundle that I mentioned before that had been observed by Fernanda Tovar-Moll and also Elliot Scher's group in people with partial agenesis. And what Tim found by tractography was indeed that mice have this ectopic projection. And he actually even replicated something that we still don't understand, that in humans and also in Tim's mouse model, this is an asymmetric projection in terms of how many streamlines seem to be produced. So sort of right to left has a lot more streamlines than left to right. And this asymmetry is also seen in people. So it appeared that, in fact, maybe mice with partial agenesis of the corpus callosum are also capable of forming these ectopic sigmoid bundles. But what's great about using mice is that, of course, we can go in and verify this result with anatomical histological techniques. And so Tim joined up with Laura Fenlon, Dr. Laura Fenlon, who's a postdoc in my lab, who is truly a master at electroporation. And Laura electroporated, introduced a TD tomato construct into the caudal region of the cortex here. And these are what you're looking at, are horizontal sections of the mouse brain. And so she did the electroporation at embryonic day 15.5, when the majority of callosal neurons are being born and migrating to layer 2-3, where the majority of these neurons reside. And what you can see in the controls is that these axons project mostly homotopically. But in the partial agenesis mice, what we find is that they do project homotopically, but they also have a projection into this rostral heterotopic region. And she measured the amount of fluorescence in these two areas and actually found a difference in that there's fewer axons going to our region of interest 2 and an increase in the number of axons going to region of interest 1. So this is at least suggestive that this sigmoid bundle does in fact exist in mice. So Tim also used graph analysis to look at whole brain connectivity in this BTBR-N2 cross. And what you're seeing here, I'll just walk you through it, is a graph analysis where he parcellates the brain into different regions and then looks at the connectivity between all those different regions. And in this kind of image, what he's showing is these blobs, these dots here, vary in size depending on the amount of connectivity coming into or out of that region. So the bigger circles have more connectivity. Another way to represent this data is to put all of the different areas of the brain on the left side and the right side around this circular plot, and then look at the connectivity between different areas of the brain. So these are just two representations showing very similar data. And now I've explained that, maybe you can see that there are some quite significant differences. So here, this is the control mice. here are mice with complete agenesis of the corpus callosum. Here are some littermate animals with partial agenesis of the corpus callosum. And here is what Tim calls the virtual callosotomy, which had also been done in humans previously in a similar kind of analysis in Elliot Scher's lab. And what you do here is you virtually, that is, remove the callosal crossing connections. So, and the reason you do this is to see, is it that if you just remove the callosal crossing connections, you would get a similar view as complete agenesis of the corpus callosum. And it's very, very different. And this suggests that, in fact, there is complete reorganization, both within the nodes. You can see many of these nodes are much larger in the complete ACC compared to the control. And they're really quite different in the partial agenesis of the corpus callosum group. So this suggests that there really is a reorganization of the ipsilateral tracts in each hemisphere as a consequence of the agenesis of the corpus callosum. So from that, hopefully I have demonstrated to you that perhaps there could be two different types of callosal wiring disorders. So midline disorders, which we can see here, they're very evident by MRI and easier to study. And this results in altered ipsilateral connectivity, which we still don't know how that really impacts function, but something that we're working on. And a second group potentially where we have normal crossing but abnormal targeting in the contralateral hemisphere. So this is a hypothesis, pure speculation at the moment, but something that we think is potentially very interesting work for the future. So I'd like to now move to just tell you a little bit about mechanisms of how the corpus callosum forms. And I've separated this into midline guidance cues and also how fibers find, how axons find their target in the contralateral hemisphere, which is driven by both molecular and activity-dependent mechanisms. So what you're seeing here, this is an image that you'll see a number of times in the rest of the talk, is a postnatal day 10 mouse brain coronal section, which was electroporated at embryonic day 15.5 with a YFP construct. And the cell bodies are up here in layer 2-3, and all the rest of the fluorescence are the axonal projections. So this was electroporated into S1, primary somatosensory cortex. So you can see the axons diving down through layer 4 in between the barrels and arborizing in layers 5 and then projecting out and across to the contralateral hemisphere, but also there is this ipsilateral projection here. So just to talk about the midline guidance cues, so Alan and many other groups in the field for decades have been working on different axon guidance mechanisms in the developing nervous system that really separate into different types of molecules and mechanisms of how growth cone target different areas of the nervous system. And we started out in this field as well and applied this to trying to understand these mechanisms in the formation of the corpus callosum. And in that work, we identified, along with many other people in the field, that glia are crucially important for wiring the brain. And there are glia around every commissure throughout the whole neuroaxis in every species that has a bilaterally symmetrical nervous system. And here you're seeing a number of glial populations that are very important for formation of the corpus callosum. So Jerry Silver's group did some seminal work on this in the 80s, and 90s and identified glia, a glial plate within this area that's very important. We identified that this plate is actually made up of a number of different populations of glia. These ones we call indusium griseum glia, and these we call the glial wedge. And we know that a family of transcription factors called the nuclear factor 1 genes are crucially important for the formation of these glia. And these are just some of the axonal guidance molecules that are expressed in this region. So on the growing tips of the growth cone are all the classic guidance receptors, ROGOs, DCCs, neuropilins, plexins, and also EPH and ephrin molecules. And their ligands are expressed by these glia or by different neuronal cells within this region. And later on, I will be talking a little bit about DCC and its ligands. So just to go back to the nuclear factor 1 genes, so we're interested in not only what these glia express, but how these glia develop, because they are so crucial for commissure formation. So I've been working on this family of transcription factors for about 2 decades, and with my wonderful collaborator, Richard Grunstowski at SUNY Buffalo. And a number of people from my group continue to work on these genes in their own labs. Mike Piper and Jens Bunt, who just recently moved to the Netherlands, and carrying on this work in my lab at the moment is Jonathan Lim. So the NF1 group of family of genes have 4 family members. 3 of these are expressed in brain, A, B, and X, and they have a very highly conserved across the family DNA binding domain. They appear to act at the interface between proliferation and differentiation by binding DNA and directly affecting transcription. But we also have evidence from our group and other groups in the field that they are very important also perhaps as epigenetic regulators. So they bind in the context of their function as transcription factors. They bind DNA and they directly affect the expression of genes such as glial fibrillary acidic protein and other differentiation genes. And they also regulate by turning off proliferation genes such as the Hess family. And so I'm not going to go through all of this work today, but suffice it to say that NF1s are very much involved in this balance between amount of proliferation and differentiation within the brain. And this then has flow-on effects, if you like, in terms of different disease and developmental phenotypes that we see. So not only are they involved in development of the corpus callosum, and I'm going to tell you how in just a moment, but they are also involved in ventricular size, and also we have implicated them very strongly in both childhood and adult brain cancer, and we've done a little bit of work on that, but I'm not going to talk about that today. So NF1 genes have been associated with callosal anomalies for quite some time, and this is a lovely paper from the Developmental Genome Anatomy Project at Harvard Medical School, and you can see different patients here with NF1A mutations have different callosal malformations. And so their role in cortical development is something that we're actively working on along with other groups in the field. They're expressed by radial glia, by astrocytes, neurons, oligodendrocytes, both during development and in the adult brain. And A, B, and X knockout mice have severe cortical phenotypes. And what they have in common is dysgenesis of the corpus callosum, hippocampal malformations, enlarged ventricles, and enlarged brains. And each of these family members have also been associated with different human developmental disorders, and the human affected people also have megalencephaly, so enlarged brain, enlarged ventricles, dysgenesis of the corpus callosum, and they have developmental delay and intellectual disability. And like many of you in the field, we're very interested to understand how developmental delay and intellectual disability come about from an enlarged brain and malformations in the wiring of the brain. So each of these, as I said, is implicated in different human developmental disorders. And this is a very nice review of these human disorders that was written and in a collaboration with Martin Zenker's group and Raoul Henkerman's group. So I'd like to turn now and just tell you about, very quickly, about the cause of complete agenesis of the corpus callosum. And this is work that Ilan Govius did when she was a postdoc in my group, together with Laura Morcombe, who has just completed her PhD and is now a postdoc at Cambridge University in the UK. So the key here, which you can see, is actually, so the fibers are failing to cross here, but what's different? And you can see that there is a major anatomical difference between the neurotypical control and the people with complete agenesis. So here, underlying the corpus callosum is a very small piece of tissue in adult humans, which is the septum pellucidum. So the septum, comparatively, is much larger in mice than adult humans. And you can see at this stage, the people with complete ACC have these open septal leaves. And so we followed this up using some of the patients in Elliot Scher's cohort. And we looked at every case that he had with complete agenesis of the corpus callosum that he had good imaging data on. And we looked at the anatomy, and we identified that in 100% of those cases, every person had open septal leaves. And this is what we call the interhemispheric fissure. So I'm going to be talking a lot about that. That's the gap between the two hemispheres. And this is vastly expanded, of course, in people with complete agenesis of the corpus callosum. So how does this come about? It's an early forebrain developmental disorder. The corpus callosum forms between 12 gestational weeks and 20 gestational weeks. And what you're seeing here is a scanning EM of the mouse brain looking front on, and you can see the interhemispheric fissure here. Across development, and the corpus callosum is going to cross around this level in the mouse, and this is in the human brain, and you can see the same kind of fissure here. So what we found looking at developing human brain is that indeed there is a population of glia that reside along the cleft here. And these start out as an open zipper. So these are known as the midline zipper glia, and they were first identified by Jerry Silver. And Jerry had a hypothesis that, in fact, the midline zipper glia, he named them the zipper glia because he hypothesized that they were crucially important for fusing the two hemispheres together. And indeed, that is the case. So at gestational week 14, you can see the zipper is now closed and there's a fused midline. So this really happens as the cells migrate towards the midline from the lamina terminalis, and they intercalate at the midline, eliminate the leptomeninges, and fuse this area of tissue, forming the bridge, which is very necessary for the formation of the corpus callosum. And here you can see an MRI of a 13-week gestation human brain. It's one brain, but different views. And you can see, looking from inferior to superior, different levels, even with within this one brain, open septal leaves haven't fused yet, but in this more inferior region, there is fusion of the septum. And you can see the septum is much larger relative to the cingulate cortex above in this fetal brain, but in the adult is very, very small. So this also happens in mouse, and these are now horizontal sections in the mouse, and what you're seeing is the hinge area here. And this is where the cells are born. This is the lamina terminalis here, the third ventricle and the two lateral ventricles. And the cells are born there, and they migrate forward along the fissure, where they then fuse and intercalate. And it's at this level here, I can show you, by labeling with in utero electroporation the very first pioneering axons that cross the corpus callosum, which come from the cingulate cortex, they actually require this region to cross the midline. And they do so. In fact, they travel down until they can find an area where there's a fused bridge of tissue that they can cross. Now, just coming back to the nuclear factor 1 genes, these mice, in both NF1A knockout and NF1B knockout, have this open interhemispheric fissure. And this is also replicated in patients with NF1A or NF1B haploinsufficiency. And what we found, in fact, was that in these mice, they have very underdeveloped midline cypiclia. So they are formed, but they don't progress to differentiate. And this whole region, so this was published some time ago, it is all driven by the morphogen fibroblast growth factor 8 and its receptors, which are expressed on the midline cypiclia. And we know that the nuclear factor 1 genes are required for driving this differentiation, because if we overexpress FGF8 by in utero electroporation, we can drive cells in this whole area to first turn on GLAST, an immature glial marker, and then to progress to GFAP positive cells. Whereas if we do the same experiment in NF1A or NF1B knockout mice, we can't drive these cells to full differentiation. And so this is the cause of complete agenesis of the corpus callosum, we believe, in both mouse and in humans, that the axons come down, they fail to cross, and they form these probe spundles if these midline cypiclia cells don't migrate forward and intercalate at the midline. So not only is FGF8 and NF1 involved in this, but more recently we have found that other axon guidance molecules that we know are important for guiding axons across the midline, but also have an earlier function in the development of midline cypiclia. And the first one is DCC. So DCC, as you know, deleted in colorectal cancer, is an important transmembrane axon guidance molecule with a number of different ligands, Draxin and Netrin. And we have evidence that all of these are required for the development of midline cypiclia, and so function at a stage just before they also function in axon guidance. So in DCC mutant mice, they fail to form commissures throughout the nervous system. And in fact, people with mutations in DCC display this phenotype, which is called congenital mirror movement disorder. And you can see this boy is trying to inhibit one hand from moving when he's moving the other hand. So normally this is not a problem, but for people with congenital mirror movement disorder, he can't stop this hand from moving, even though he's just trying to move one hand. And DCC mutations were identified in human populations initially by Guy Rouleau in Canada and Emmanuel Rose's lab in France. And they identified that DCC was related to congenital mirror movement disorder. And but the people that they identified initially had a full corpus callosum, and this was really quite confusing for some time. But our group and their groups, and Alan involved as well, has identified that, in fact, DCC is required for formation of the corpus callosum in people and in animal models as well. And there are a huge number of different mutations that have been identified, even more than these now, across DCC. And the patients either have just the congenital mirror movement disorder or congenital mirror movement disorder with callosal anomalies, or both. And this was published by our consortium, but also by Chris Walsh's group at the same time, and has been followed up by a number of other groups. So this also occurs in the mice and has the same phenotype. So in Netrin-1 hypermorph mice, DCC knockouts, and also the Kanga mice all have open interhemispheric fissure. And I won't go through, we have a paper on the bioarchive at the moment, but DCC seems to regulate the development of these midline zipperglia, in particular their migration and intercalation at the midline, and their ability to differentiate into mature astrocytes, which are required for the intercalation step. And just moving on from this work, we are also actively investigating how partial corpus callosum dysgenesis, or partial agenesis of the corpus callosum, occurs. And we're using this BTBR-N2 model. And we have another paper on the bioarchive on this, where I remind you that this N2 cross has this distribution of commissural size. And together with Elliot Scher's group, we've used this linkage analysis and identified a quantitative trait loci on chromosome 4, which we've identified as being a truncation of Draxin in the BTBR knockout mice. And I don't have time to go through that data, but Draxin is also very important, as well as Netrin, for the development of these midline zipperglia. And in fact, Draxin, the del-del BTBR mice, but also the BTBR single line have a similar midline zipperglia defect, in which there are more midline zipperglia progenitors produced. There's also an increase in proliferation in the leptomeninges, which expands the interhemispheric fissure. And there's a disruption of radial glial processes. Their migration is, they over-migrate the cells in these mice, and there's a failure to intercalate. So our data thus far has identified 6 steps in the development of interhemispheric fissure remodeling. This proliferation of these midline zipperglia cells, their specification as midline zipperglia cells, they need to extend processes, anchor them as radial glia, both to the apical and basal sides, and then somally translocate to the midline, where they then differentiate into multipolar astrocytes and intercalate across the fissure. And this elimination of the leptomeninges, we're still not sure how this is occurring. But once this tissue intercalation is complete, then callosal axons use this intercalated glial bridge to cross the midline. So in the last few minutes, I wanted to talk a bit about activity-dependent mechanisms of corpus callosum formation. And previously, some work from our group used the mouse somatosensory system in order to test some hypotheses on this. And this system is great because it's a fully cross projection. From one whisker pad on one side of the face, the information is topographically projected into the cortex. And it's at the level of the corpus callosum where the first integration of information between the 2 hemispheres occurs. And Rodrigo Suarez and Laura Fenlon in my group asked what would happen if we cauterized the whisker pad in mice. How would this affect the development of the corpus callosum? And we were inspired by work that had been done by Chun-Li Wang when he was at the Institute of Neuroscience and in Shanghai, and followed up on this work to look at how bilateral cauterization of the whisker pads might affect callosal formation. And since this is published, I really won't go through it in much detail. But what we found was that similarly to what had been published previously, there was a dropout of callosal axons in unilaterally cauterized mice. But we were very surprised when we performed the bilateral cauterization and found a rescue of this phenotype. And this did not occur in projections that were going into either S1 or into this insular perirhinal region, and was really restricted to this area, which is at the S1, S2 border. And so we know that this is actually, we were very interested to look at whether this had any, this was a result of some asymmetry in the amount of activity coming in, which maybe then was symmetrical when we removed both whisker pads. And so we removed whiskers in different configurations, either asymmetrically or symmetrically on the face, and we saw a partial rescue when we did this symmetrically. And so we also identified, working with Pankaj Shah's lab, that these were functional connections, and so this is the rescue here, this is the control, the unilateral and the bilateral cauterization. And so there was this morphological rescue of the bilaterally cauterized animals represented this persistent functional rescue of callosal wiring. And so more recently, we've been looking at how activity actually develops in the fat-tailed dunnart. And one of the reasons that we moved to this animal model is because the pups are born into the pouch at an extremely early developmental stage. So here is a developmental stage, and here is mouse embryonic day. So here's mouse birth at P0 at equivalent of stage 27. So, and this is at equivalent dunnart postnatal day 29. So they are born at a very, very underdeveloped state here at stage 18, which is equivalent to embryonic 10 in the mouse or a 7-week gestation human. And all of the development of their cerebral cortex, since this is virtually not too long after neural tube closure, occurs in the pouch. And this work has really been led and championed in my group by Dr. Rodrigo Suarez, who's really an outstanding scientist and has developed this model to investigate both evolution of the brain, of the cerebral cortex and its commissures, but also to do this work on looking at activity-dependent development. So here's what a dunnart looks like when it first develops, when it's first born, very underdeveloped. And so Rodrigo, together with colleagues in the lab, has developed ways of doing in-pouch electroporation, so we can manipulate gene expression, introduce traces and different fluorophores into this animal. And you can see here their 6-layered cerebral cortex. And what's the incredible advantage of this model is that you can perform multiple rounds of electroporation in the same pup at different stages quite easily, because you're not having to do a full laparotomy and in utero electroporation that we need to do in mice. Another great advantage of this animal is that at early stages, they have a transparent skull. And so we've been able to do 2-photon calcium imaging directly through the skull. So Rodrigo has been working with Toby Blewett, who was a master's student. He's now just graduated, to perform calcium imaging using GCamp Success in the fat-tailed dunnart. And what Toby has been able, so everything had to be set up in this species because it had never been used before for anything like this. So Rodrigo has developed a staging method so we can very accurately stage these animals. He's established all of the electroporation techniques and also the ability to do this calcium imaging together with Toby. And so to analyse this kind of calcium imaging data, we've collaborated with Jeff Goodhill's lab, and Lilac Aberton began this work where they have been using some very sophisticated methods to model the calcium patterns of activity in the brain. And this work has really been now led for the last 2 years by Michael McCulloch, who's been applying some sophisticated statistical analyses to this calcium imaging data together with Jeff. And I'll talk a little bit about those. So the developing cerebral cortex, it has been known for some time that there are early patterns of activity that occur in the sensory areas. So this wonderful work from Hidenobu Mizuno, together with his colleagues, Shingo Nakazawa and also Takuji Iwasato, and have been looking at and identified patchwork type activity in somatosensory cortex. And also in Michael Craer's lab, James Ackman has identified these beautiful travelling waves of activity in V1. And so we were interested to know, are there patterns of cortical activity in the developing dunnart sensory areas? And we identified that there are patchwork type activity in dunnart somatosensory cortex. And Michael has applied graph analysis and a number of different statistical techniques to look at how the activity is correlated across this patch of cortex. And we've also identified that in V1, there are travelling waves. And interestingly, we also see, so this is the same patch of visual cortex and the same imaging session, but we see different directions, perhaps different patterns of these travelling waves. And Michael has been applying different techniques to look at the probability of these waves travelling a certain distance and the direction of their travel. And this is called a Windrow's plot, which shows you the probability of these activity events and the direction of their travel and also their distance, so between 0 and 14 to 1600 microns. And so one of the questions that we wanted to ask was when does the brain first switch these activity patterns on? And so in somatosensory cortex, we electroporated the animals at postnatal day 10, this very early stage, and we began imaging initially at these older stages and then went earlier and earlier. And so we can see patchwork type activity at stage 25, 26 and 27. We can see events even at stage 24 and even at stage 23. And in the visual cortex, we can see these correlated waves of activity at stage 26 and even at stage 25, but not earlier than that. And so some of the analysis that Michael has just done, and this is not published yet, has been to look very carefully at the statistics of this correlation. And what we find is that, so this is a sort of quantitative analysis of that, that data that I was just showing you, was that at stage, so this is stage 23, 24, and 25, that the correlation between different areas of the network increases with developmental stage, and there's more highly correlated activity when the voxels are closer together. And so this correlation for less than 200 microns increases with developmental stage. And this is sort of a schematic representation of the correlation of nodes of activity within S1 that occur over development. And you can see patches begin to really emerge. It's a little bit at stage 23, but the patches really begin to emerge, and they're color-coded here based on the amount of correlation that they have. And he's quantified so these are called assemblies. So the number of assemblies increases with developmental stage, and the number of the nodes that participate in these assemblies increases, and the strength of these nodes also increases with development. And for V1, these Windrose plots also demonstrate, so you can see they're color-coded here. So these are not traveling very far. These are between 0 to 200 microns at stage 25. But over time, we have more waves and they're traveling further. And this is further quantified here. So these are these event durations which increase over time, the distance increases, and the area of these waves increases, and their frequency over time. So there does seem to be a slightly delayed development of these patterns of activity in V1 compared to S1. And these are some recordings from the same animal in the same recording session. And if we take all of these different measurements and perform some multidimensional scaling on them and pull stages 26 and 27, the patterns of activity in V1 and S1 really separate in an unbiased way into two different groups. So there does seem to be quite different activity in S1 versus V1. And so we're starting to think about when and how does the brain really switch on. These activity patterns in S1 and V1 of the cerebral cortex, they are evolutionarily conserved. We know that now between mouse and the Dunnart, that the Dunnart cerebral cortex develops these patterns of activity from stage 23 in S1 and stage 25 in V1. And these are the mouse equivalent days. And these patterns of activity emerge from the outset. It's not that you have activity everywhere and then it becomes specialized. These patchwork-like activity and the wave-like activity emerge from the outset in these different sensory areas. So hopefully today I've shown you that both cellular and molecular mechanisms and activity-dependent mechanisms are all extremely important for formation of the long-range projections of the brain, and that these really underpin function, and this is an area that we really need to do a lot more work in. And I'd like to thank the members of my group that have been involved in the study. I mentioned many people throughout the talk. And as Alain mentioned, well, I should also first thank the families of the Australian Disorders of the Corpus callosum group that I work very closely with. As Alain mentioned, I am moving to the University of Washington in St. Louis to take up the role as chair of the Department of Neuroscience there, as well as the director of the McDonald Center for Cellular Neuroscience, and I hope that I get to see you all in St. Louis sometime and can invite you to come and visit our department. Thank you very much.
Thank you, Linda. It was great. So you can stop sharing your screen and then we can I can read the questions that you got.
Thank you.
Okay, so first question, there are two questions about the sigmoid bundles. So first is from Eric Mir. Do you know if sigmoid bundles only arise from callosal neurons, or can other types of cortical neurons contribute? Could it be linked to broader defects in brain connectivity?
Yeah, so we have only studied the layer 2-3 neurons with regard to, so you certainly can't study this by tractography and MRI. There's just not enough resolution. And the only work that we've done in the mouse was by electroporation, where we labeled layer 2-3 neurons. So that's all I can say about that. And the second part of the question was...
could it be linked to broader defect in brain connectivity? And then...
yeah, so there's some suggestion, although really we need a lot more participants and we need to do more cognitive analysis. But there is some suggestion that these ectopic bundles, the sigmoid bundle, may actually not confer an advantage to the person cognitively. So they're more severely affected, people with this sigmoid bundle. So it's not always that these ectopic projections are actually, you know, conferring a better cognitive outcome.
Okay, thank you. Then second question from Marta Nieto is, have you looked at how the sigmoid bundle arises during development, for example, by looking at earlier postnatal stages? So when do you first see the evidence for a sigmoid bundle, if you image like kids or baby that have malformation, I guess?
Right, yeah. So this work has not been done in really young children at all. So I wouldn't be able to answer that.
Okay.
I don't know.
Okay.
Yeah.
Then
I'm assuming that occurs very early in development.
Okay, then a question from Hernan Lopez here. Would you be interested in commenting about the somewhat controversial sexual dimorphism in the corpus callosum, and whether, if it indeed exists, is of any physiological relevance?
Yeah. So I think what's being referred to here is the sagittal size of the corpus callosum by T1 imaging. So this is structural imaging. But really, I think a lot of that work needs to be a little bit revisited, because as I've shown you in my talk, just seeing a T1 weighted anatomical image, mid-sagittal view, tells you nothing about the connectivity of the corpus callosum. It might tell you that relatively there may be fewer fibers crossing, but it doesn't tell you where they're going or even how many there are. And so so this old work was really looking at just tracing around the corpus callosum in males and females and saw a difference. But the impact of that is really, it's not known.
Okay, thank you. So then another question from Thomas Hummel. Do ectopic heterotopic commissures follow existing fiber bundles or build de novo tracks?
Say that again, sorry.
Do ectopic heterotopic commissures follow existing fiber bundles or build de novo tracks?
Oh, well, there's evidence in mice for all of those. So many of the early knockout mice that we've all studied in axon guidance molecules, some of them have ectopic tracks in the forebrain and even ectopic commissures. So it's possible, but in humans, certainly, we very rarely see ectopic bundles crossing the midline. So for example, in BTBR, there is also another ectopic kind of crossing that occurs in the thalamus. that's been identified. But it's not been replicated in every lab. So I would say that there's a lot of axonal plasticity possible, and that there aren't just immediately other tracks that are formed. And it really all depends on what the gene is doing in other areas as well. So a gene involved in axon guidance might also be involved in other developmental events within the brain.
Thank you. So another question from Kim Bashave: is the asymmetry in ACC always on the same side?
Yeah, so it's often so I think maybe you're referring to the sigmoid bundle.
I guess. Yeah.
So it's often very similar in across different patients, that it's stronger from right to left. And that is, I would love to hear if anybody has any ideas about why they think that might occur, but it does seem to be quite consistent. Although, trying to get enough participants so that you can really have, a big sample size, that's our goal. And, we need to be able to do the same kind of experiment across a large number of participants to be, more certain about this.
Okay, thank you. So next question again from Marta Nieto. Are the mirror movements of DCC patients with a CC related to abnormal targeting of CC connections, or are they related to other mechanisms?
Well, would you like to answer that, Len?
So they are. Yeah, it's related to other mechanisms. It's not clear yet. I mean, there might be some CC problem, but it's more related to probably even spinal cord circuits, etc. Also CST anomalies, but it's not 100% sure, but probably not so much the CC defect.
Right.
Yeah.
So I think Alain and I and other people working in this field would all agree that the mirror movement disorder is unlikely to be related to the corpus callosum malformation, and is more likely to be related to other wiring defects of descending projections and missing at the pyramidal decussation, but also the spinal motor circuits as well.
Okay. So then a question for Eikmir again. So could you speculate on the elimination of leptomeninges at the fissure? Is it active elimination via contribution of immune cells, for example, on apoptosis?
Yeah, so we don't know enough about how the leptomeninges is eliminated. We tried looking at this previously, and we identified that the midline Zippiclia express matrix metalloproteases that may be involved, but we actually don't know how the leptomeninges is eliminated, and we do think it's probably an active elimination of the leptomeninges. But it's a great question, and we need to understand it.
Okay, so then a question from Rashmit Kaur. Abnormal contralateral targeting is always activity dependent? Question. And why several mutations on one gene produces heterogeneity? I guess is why. Yeah, why do you have a heterogeneous, like for the DCC, for instance, phenotype, or for the BTBR, like you always have some. It's the same mutation, but you have a spectrum of.
Oh, right. Right.
So is it activity dependent with contralateral targeting, and then?
Yeah. Okay. So let me just say that the BTBR mouse, we identified a number of different loci that may be contributing across the genome. So Draxin is definitely contributing, but there's going to be other modifiers that are also contributing that we haven't identified the actual gene.
Yeah, because the Draxin knockout, I think, has no commissures, and all the mutants have no commissures always. It's not like random or variable.
Yeah, but this was a question about the BTBR, wasn't it?
No, it
And-
was about the why several mutations of the same gene produces a heterogeneous phenotype.
Right. So maybe what I was talking about is the BTBR mice, right, which have, they do have this mutation in Draxin, but we think there's also other modifiers. Maybe I could address this with the DCC. So DCC people have very heterogeneous phenotypes, very heterogeneous phenotypes. It's quite a different thing in humans than in mice. So, you know, in mice, we're making major deletions, knockouts. In humans, they're usually point mutations, because if they had a major deletion or a knockout of DCC homozygote, the fetus probably would not survive, or the baby wouldn't survive. So, so most of the human mutations we see are point mutations that have different effects on the protein, and maybe the binding and signaling. So it's you know, those are going to result in heterogeneous phenotypes. So maybe I'm not answering the question correctly.
No, I think it is. Yeah, I think good answer. Then we have a question then again on the sigmoid bundle. So does the sigmoid bundle always project to the same region? Do you think there are early problems in arealization that allow projection of those neurons to regions that they are not supposed to go normally?
An interesting idea. You know,
Effil. I don't know who that is. It's from Effil, but I don't have the last name.
Okay. Well, it's a very good question, and a very interesting idea about patterning of the cortex and whether that has an impact on how this ectopic projection forms. We don't have any data. I should say that if anybody wants to try this experiment of crossing the B2BR with the C57 Black 6, I mean, you can look at our data. The number of partial ACC animals you get from that cross is not high. So the way that Tim had to do this experiment was cross many litters, generate many litters, and then he did a quick, very short MRI scan on about 150 brains, and of course keep the DNA from every animal, before he could identify a cohort of 10, which he could then work with. So it's actually hard to get large numbers of animals with partial agenesis of the corpus callosum.
Okay.
And unfortunately, in most knockout mice, they have complete ACC instead of partial ACC.
Okay. So I think the last question for me, and then we're gonna need to close. I told you, I promise you we won't stop when there will be only the two of us left.
Okay.
So now in terms of, so the way I understand your talk is, at the end, is there any evidence that callosal crossing defect is due to axon guidance problem? It seems that, I mean, per se, like the way you present it is always midline problem, and at the end, is there any axon guidance receptor whose mutation could cause a causal defect, or it's always secondary problem? So it's non-cell autonomous.
I think that there's room for all in this. You know, there's so much overwhelming evidence that axons, callosal axons, are guided by axon guidance molecules. So we know that they use axon guidance molecules to cross the midline, and we know that they use them for targeting. So I think that they're required for different stages. I think that, you know, because they have this early effect doesn't mean that they don't have the later effect. Does that make sense?
No. But it's just to be striking that each time it's related more to like a midline fusion problem. So it's a bit.
Yeah.
Yeah. Anyway, it could be related. Maybe the axons also influence themselves, the closure, something like that. Right. There's a possibility that the axons actually influence the development
the midline septalia indirectly or something like that. Certainly the timing of this is, that's potentially possible for sure. But I don't think our data is pointing to, a complete revision of the role of DCC or anything like that. I think it's just showing that there is this earlier role in glial development. And then on top of that, DCC also acts in axonal guidance. And as I said, remember, they have to find their target in the contralateral hemisphere. And although we know that patterning is extremely important for that, we know that activity is extremely important. Those have to be read out in terms of guidance molecules, most likely. So
I think we should stop here.
Not dead.
It was a great talk for the Women in Science Day, I think. So perfect match. and then have a good dinner now. And then we'll meet again on Worldwide Neuro next week for Nenad Sestan. So again, some cortex. And thanks everyone for staying up until the end. Okay, bye-bye, Linda.
Thank you very much.
Bye.
Bye-bye.